Um, First, I want to thank the organizers for inviting me to give this presentation. It's a really nice opportunity, and it's very exciting to see that the RNA puzzle community is connected to the CASP community now. So let me start with thanking my postdoc, two highly talented postdocs who contribute to this project, CASP15 Structure Prediction Project. And so the calculations were performed by Dr. Jun Li and Dr. Sichen Zhang, and Jun did the 3D part and Sichen did the 2D part. So as you can see, my project, approach has 2D and 3D separated, kind of. So we used, I call it a hierarchical and multi-scale RNA structure prediction method. It's, as you can see, it's not a machine learning method. And the basic strategy is to first get a 2D structure, or in protein community, we call it contact map from the sequence. Then based on the predicted 2D structure to predict 3D structure. And it's a combination of a series of different models developed in my lab over the years. For the prediction from the sequence to the 2D structure, as I show here, we use the sequence information, sequence aligned MSA. And if there's some available experimental information, we'll make use of them, like a shape. In the RNA puzzle, thanks to Richard Das' lab, often the shape data was provided. And then, based on the constraints, if possible, if available, based on the sequence analysis and experiment data, we run our physical model. It's called a V42D, which I will very briefly tell you the key idea of this model. And it's available, we have a server. And basically in this V42D model, we developed a free energy landscape from the sequence. It's a pure physical model. And the key part of this model is to calculate free energies for a given structure. For example, you have this three-way junction structure. We want to calculate loop free energy. So the way we did this calculation is we sample all the conformations based on the coarse-grained model. And in this coarse-grained model, we represent a nucleotide with two or three virtual bonds. Like in the early days, Wilma Olsen's virtual bond in DNA structures. And then, based on the statistical frequency data, we call it the Ramachandran plot in protein business. And based on this data, we did Monte Carlo sampling to sample the structure, the conformation. And from the sampling, we calculated the probability to form this, to fold into this structure. Then we converted that into the effective free energy of these different motifs, like three-way junction, pseudoknots, and even the kissing loops. So that's the the physical model, V4 model, V4 2D model. And as I also mentioned, if there are available sequence information and experimental data, we use those information as constraints. For example, the shape data, from shape we can get the information about, at least we know which part is in the not in the loop, not in the helix at least. And if the sequence information, we can, there's some sequence data available, and we get a structure, some constraints from the base pair information from RFM. And if not, we sometimes build our own MSA to get to the from covariation, and we get to the base pairs, those conserved base pairs, and use those as a constraint in our physical modeling. And also for larger structures in this CASP15, we have 600 nucleus, these big structures. We through visual inspection, not of course not through eyes. When I say visual inspection, I mean through sequence comparison. We ran script to search for the sequence complementarity. Then we make them make long range contact for this CASP targets. And once we get the 2D information, then we run the 3D structure prediction. We have three main method models to run the 3D structure prediction from the 2D. So one is purely template-based. If we can find a template, then we use a template-based model. It's we call it V for the 3D and V for the LA. So let me very briefly tell you what these models are. So for V for the 3D, that's our first generation oldest model for 3D structure prediction, basically. If you get a 2D structure like this is like a tRNA kind of structure. It's a four-way junction. And look, the key part is to get the 3D structure for this central loop. So you go to this central loop, we found that it's 0, 2, 1, 4. There's a 1 nucleotide, 4 nucleotides. Then you go to the PDB database, you get all the four-way junctions and search for these combinations. And from sequence comparison, and you find the one that's closest to this sequence. That's a structure. So that's the basic idea. But often we know that the RNA has a very limited database, so often we failed. Then we go to the next method. It's called V for LA. LA means loop assembly. So basically, instead of so greedy by searching for the whole motif template, we go to each loop, each strand. Based on the sequence similarity, we found the good template. They assemble them together. That's what we call the loop assembly. So the good thing that for this strategy is that we can have high success rate of find the template, but the disadvantage for this kind of approach is that often when you assemble it up, I mean, it's not a good structure because you lose the information of this whole motif. Okay, good. Then we have another method. What if you failed? So we calculate the structure, the missing pieces. We use an MD simulation method developed in my lab. We use a coarse grain, we call it pseudo atom-based molecular dynamic simulation method. So in this method, basically, we use a coarse grain model, which you can see with one bead for the phosphate, one bead for the sugar, and you have 3 or 4 or 2 beads for the purine pyrimidine. And based on the known structure data, we developed the force field, the coarse grain force field, such that the simulated conformation distribution agrees with the observed distribution from the PDB. Okay. Recently, similarly, following this similar MD simulation strategy, we developed another model called RNA junction prediction. I won't spend too much time on this model. And this model is not published yet, but it's really a cool model. Two key advantages of this model is that it can sample the global topologies of, for example, this is three-way junction. You can sample global topology of this helix orientation, different combination of the base stack of the helical coaxial stacking. And it can also sample the loop-loop interactions and those non-canonical interactions. So these are the toolbox of our methods. So next, I'm going to very quickly go through some of the showcases. I'm going to talk about the two not so bad or successful prediction and two failed predictions. This target, R1126, is a synthetic designed RNA. And this is a structure on the left is a 2D structure we predicted from our V4 of the physical model without using any sequence alignment. So we almost got it right, except this part. On the right is the native structure, 2D structure for the native fold. And this part is a G quadruplex, but in our model, we cannot handle G quadruplex. So what we did was after we predicted this 2D structure, we went to the PDB database, we did a sequence alignment, and we found this local alignment fit this G quadruplex. So we are sure this part should be the G quadruplex. So we put the G quadruplex here. Then, as I said, we compare the sequence. We found this loop here and this bodge here. They can form perfect sequence complementarity. So we believe probably they form kissing base pairs. So we added these kissing pairs. So these are the two things we did after the physical modeling. Then we did the 3D MD simulation, and we used the template-based structure, the loop assembly method, to build the initial structure. Then we ran the MD simulations, and this is the same 2D structure as the one I showed. It just shows different ways. And the final structure looks like this, and the RMSD is 12.6. It's not perfect, but given the size of the structure, it's not too bad. We think the success story for this target is that the 2D structure is accurate. The 2D structure is accurate because first, the good physical model, and the second, we use the sequence alignment to identify the G quadruplex. Otherwise, we will never be able to find this G quadruplex. Okay, so the correct structure shown is like this, native structure. It's the kissing between the hairpin loop and this bodge loop. We actually submit another structure. We submitted this collector one. We also submitted an additional structure. That's the structure. When we got a sequence, we figured it must be designed. So immediately we thought maybe you can make this as a tile kind of thing. So it's the kissing between the previous hairpin, and the next bodge. And that's the 3D structure we built. We submitted one such models. And we still believe this structure could possibly happen. Maybe someday experimentalists can make this structure by taking higher concentration of the RNAs, and this structure might form still this crystal structure, actually. Very beautiful structure. So next, I'm going to quickly go through another case which Rachel explicitly wanted me to talk about. It's a simple ribose switch. It's a pre-Q1 ribose switch structure. So the sequence is short. It's pretty short. And the first thing we did, we went to the sequence comparison. We found two good templates here. I won't go to details. This is one template, the other template. And basically, we took the template structure, these brown color ones, and are actually similar to this template. You see, the sequence is identical for this part, and for this part, it's quite a similar sequences. We just took the PDB structure for this part, this brown, these three strands, that's the structure. And we found that it turns out that the template structure and the native structure, they're almost already pretty close, only 1.78 edit structure for this template. And then if you compare the two sequences, we found that in the template you have this UUAU, and while in our R1117 it's only C. So what we did is we chop off this part and put it in one cytosine here, and we ran our MD simulation to relax the structure. So that's how we got our prediction for it. So that's the structure we predicted, and with RMSD 2.0, so it's pretty good. Finally, let me very quickly go through the two failed cases. So 1116, and on the left is our predicted 2D structure, just no sequence alignment, just the physical model. For the 2D model, that's the one we got. On the right is the native 2D structure. They're almost identical, except this base pair. In the native structure, this base pair is formed, but while in our prediction, this is opened. This is open, and this is formed. While, but here this is open. So that's the only tiny small difference. And this structure was completely failed. Let me tell you why it was completely failed. Because of the first thing we did, we want to know what this structure is, the biological function of this structure, of this sequence. So we identified that it's actually poliovirus type 1. It belongs to this family of viruses. And all these rest viruses shows us almost the same kind of 2D structures, four-way junction with the internal loop here. And for one of them, there was experimental structure. This is from Sachs experiment. And the thing that from the Sachs experiment for this structure, this is actually the RV14 structure published in RNA, and show the coaxial stacking between the A and B and the C and D, the four helices, the coaxial stacking. That's the Sachs data shows. So we took that data. We blindly trusted the experiment data. So in our prediction, we force A and B, C and D to co-align. And while in the native structure, correct structure, actually they don't. It's the other way, A and D, B and C. So that's why we screwed up this whole prediction. And turns out, when we submitted models, we ran our physical model. It's RNA-JP. This is the new model, which is not published yet. This model, if we forget about experimental data, we just run our physical model. That's the RMSD we got 7.6, and we got a correct topology. So it's a good lesson for us. I think maybe this Sachs data gives the wrong structure, or maybe they don't belong to the same family. Okay, so let me skip this. And finally, because my time is up, so finally, I will skip this and leave this to the afternoon discussion and some stories, some lessons of success and the failed stories. And finally, let me thank my generations of postdocs and students who contributed to building these models, series of different generations of models that contributed to these CASP predictions. And finally, thanks the funding agencies. Thank you all for your attention.